Bom dia a todos e todas, sejam todos bem-vindos e bem-vindas a mais um webinário internacional da Unifesp. Hoje, um webinário print com o representante print, o professor Alexandre. E em respeito à nossa convidada, o webinário de hoje será feito em inglês. So, welcome everyone for being here today with us. It's a great pleasure to introduce our guest, Professor Kathryn Lilley. Now she's in a lovely city in the UK, Cambridge, and she's from the Department of Biochemistry, University of Cambridge. And she's going to talk a little bit about the interrogating the subcellular organization of the transcriptome and proteome. But I would like also to thank you, Professor Alexandre. Professor Alexandre Tashima. He's graduated and PhD in chemical engineering by the State University of Campinas. He's professor, he got a postdoctorate at the Instituto Butantan and ICB USP. He has experience in proteomics, peptidomics, and mass spectrometry, and also in phase equilibrium thermodynamics, with an emphasis on biomolecules, volatile electrolytes, and supercritical state systems. He did a sandwich doctorate at the Delft University of Technology in Netherlands in biotechnology process and a postdoctorate in proteomics at the Institute of Biomedicina de Valencia in Spain. Professor Alexandre was adjunct professor at the Department of Exact and Earth Sciences at the Federal University of São Paulo in Diademos campus between 2009 and 2014. And in 2014, he joined as adjunct professor at the Department of Biochemistry at the Escola Paulista de Medicina. And since 2017, he has been an associated professor at the Department of Biochemistry at the Escola Paulista of Medicina. So on behalf of the Graduate Studies and Research Office, I would like to thank you, Professor Kathryn, for being here and providing this speak today and sharing your knowledge. And it's a great pleasure to have a distinguished researcher like you are here with us. And thank you, Professor Alexandre, for being a partner and organize and working hard behind the scenes to give the opportunity for our community to be in touch with Professor Kathryn Lilley. So thank you so much.
Okay, thank you, Professor Camilo, and thanks for the support team of the Federal University of São Paulo for setting up this webinar. We are pleased to have here today Professor Kathryn Lilley. Thank you for accepting our invitation for this webinar. And Professor Kathryn is Bachelor of Science in Biochemistry from the University of Sheffield and PhD in Biochemistry from the same university. She is Professor of Cellular Dynamics in the Department of Biochemistry of the University of Cambridge in UK, where she directs the Cambridge Center for Proteomics. She is also a member of the Milner Therapeutics Institute. Her research program creates and applies technologies to measure the dynamics of the transcriptome and proteome in high throughput in space and time during critical cellular processes. Her group has also contributed many open source informatics tools to efficiently mine and visualize complex data which is produced by spatial-temporal proteomics. In 2017, she was the recipient of the Juan Pablo Albar Proteomic Pioneer Award from the European Proteomics Association, and in 2018, she received the HUPO Distinguished Achievements in Proteomics Award. Not only Kathryn is an amazing researcher, but she's also an amazing person. She hosted our PhD student, Erica, during the hard times of the pandemics, and her support has been very important for her during that hard times. Before we begin this webinar, let me just tell you the format of the webinar. So Kathryn will present the webinar, and the public will be able to ask questions by the chat, by writing in the chat. And then I will ask the questions for Kathryn in the end of the presentation. If you want to send in Portuguese and I can translate, or if you prefer to send in English, please do how you prefer. Well, so thank you, Kathryn, and welcome to this webinar and to the Federal University of São Paulo.
Thank you very much. That's an extremely kind introduction. So I'm going to talk today about the technology development that my lab has been undertaking for many, many years now. And we do this to answer what we consider to be some very important cell biology questions. So just to start off, we all know that proteins have to get to the right part of the cell to carry out their job. And that when this goes wrong, this can underpin many diseases, from cancers to various neurological diseases. And if we think about where proteins end up in cells, it's dictated by many different factors. It's dictated by the sequence of the protein. Proteins may have signal sequences that tell them that they're going to be mitochondrial, and will get them through the mitochondrial import sequence machinery. It could also be post-translationally modified changes that make a protein go to a different part of the cell. Perhaps when it's phosphorylated, it will traffic differently, and also post-transcriptional modifications. So if a protein is one particular isoform, it may be in a different part of the cell to a different alternatively spliced isoform. Where a protein ends up will also be dictated by the interactions that it makes with other proteins and RNA and other biomolecules, and also where it's translated. Maybe proteins are translated in multiple parts of the cell, and that then dictates their final resting place. And finally, it may be down to conformation, that if a protein can adopt multiple different conformations, this may change how a protein is trafficked and what proteins and other molecules it can interact with. And we all know that this can completely change when you do something to cells. So if they're stressed, if there's a change in nutrient status, or even the different time of the day or different part of the cell cycle, this will lead to subcellular relocalisation. So the research questions that we have in my lab are really surrounding how we can determine where proteins are in cells and how this changes dependent on what they're interacting with and what environment the cell finds itself in. So we want to know how proteins end up in the right place. We want to know what happens if they're different isoforms, different proteoforms, different post-translational processing, what impact this has on where a protein is going to end up. If a protein makes a different interaction, where does the protein end up? Do proteins adopt different structures in different parts of the cell? Can they moonlight? Can they have different functions in different parts of the cell? And also we're interested in what role RNA plays in this. So partly through RNA-protein interactions, but also localized translation. And ultimately, we would like to know the spatial relationship between a protein and its corresponding RNA. Because as I said before, where this goes sadly wrong, and sometimes this is down to aberrant RNA trafficking, a protein will end up in an aberrant part of the cell, and this will underlie a disease. So I'm not going to talk about all of this because these are quite broad questions and quite ambitious as well. So I'm just going to take a few of these and then talk about the technologies that we've developed to be able to answer them, and to give some examples of different biological processes that we've looked at using our methods. And something that I just want to highlight, which is a very broad topic in proteomics and something which is not necessarily thought about a lot, and that's the concept of moonlighting. So the idea of protein moonlighting has been around for a few years, but was really kicked into touch about 20 years ago now by Constance Jeffries from the States. And she coined the phrase moonlighting. And in her definition of protein moonlighting is that proteins which are otherwise identical, so are not formed from alternative splicing, but identical proteins that can have completely separate functions would be described as a moonlighting protein. So a moonlighting protein would be the same protein that potentially in one part of the cell would be, for example, a dehydrogenase, and another part of the cell may be an RNA binding protein. So this idea of protein moonlighting has led to the creation of various moonlighting databases, two of which, MoonProt and Multitask, take empirical data. MoonDB does not. It finds what it considers to be moonlighting proteins by looking at nodes in multi-protein complexes. So if a protein is present in more than one of these, then perhaps that would suggest that it is a moonlighting protein. So I had a student over the summer called Lorenzo Manis from Italy, and he just had a look in the moonlighting databases, the human ones, to work out what sorts of proteins might have been shown to have different jobs in different parts of the cell. And he came up with two very alarming results that I wanted to share with you. One is that the overlap between these databases is absolutely minimal. So even when we have evidence that a protein can have two different jobs, then there's so little work that's been done on this type of thought about proteins that the databases have got very little overlap between them. The second thing that Lorenzo uncovered was what features define moonlighting proteins. So the features that he looked at was the size of the protein, whether it's involved in disease, how many interacting partners it has, where it is in the cell, all these different sort of physical features that you might imagine would dictate a protein that could have more than one job. And the only, or rather the major feature which defines moonlighting proteins is simply the length of time we've been working on them. So to my mind, this is very disturbing, because it might mean that a lot of the protein can multitask. So a lot of the proteome can be repurposed. And in our studies, we don't really think about this. We tend to think that one gene is going to lead to one protein, which is going to have one function. And unless we look at this more and more, it's going to be very difficult to link genotype to phenotype, because we really don't know what the human proteome is capable of. So that was just an aside, but the sort of take-home message from this is the overarching property that moonlighting proteins have is simply the amount of time that we've been working on them. So therefore, we need some pretty holistic methods to try and pinpoint proteins that may have multiple functions, that we can go in and then study their functionalities with different methods. So to go back to the methods that my lab have developed over the years, the first one answers the question of can we achieve an intracellular map of the proteome? And a follow-up question to that is, how do we make functional sense of protein localization? So I just wanted to highlight a paper that my student Josie Christopher and I got together earlier this year with groups around the world who are experts in protein subcellular localization. Now, they all go about it in different ways, and we wrote this very comprehensive review of the different ways in which you can find out where proteins live using very different methods, depending on what your question is. So to just really summarize the findings or what we put in that review, the way that you would find out where a protein is depends on what your question is. So if your question is, I have a favorite protein, I want to know where it is, then you're likely to use the microscopy method. So this could be through live cell imaging or immunocytochemistry. So here you know what the protein is. You either tag it with a fluorescent fusion protein, or you buy yourself a fluorescent antibody, and away you go. So that's going to tell you information on a single protein. Maybe you're interested in a particular subcellular niche. So here your question may be, I'm very interested in the Golgi apparatus. I wonder how many proteins are in the Golgi apparatus. What is the parts list of that organelle? Well, some organelles you can purify, but actually very few, because most organelles have got are very fluid and have got many overlapping physical properties. Or you could tag, or you could use proximity tagging. So these methods are very popular. And the idea here is that you would send an enzyme with some sort of signal on it to make sure that it gets to the part of the cell that you're interested in. That enzyme, by one mechanism or another, will activate biotin, and that biotin will then biotinylate any protein which is in the vicinity of where your target protein has gone. And then you simply pull out those proteins based on the affinity of biotin for streptavidin. So proximity tagging methods tend to make use of APEX, which is a ascorbate peroxidase enzyme, or a mutated biotin ligase, a promiscuous biotin ligase. So these methods are great for telling you what's in your subcellular niche of interest, but not so good at telling you if some of those proteins were in different parts of the cell, you wouldn't know what parts of the cell there were. So if proteins are in multiple places, you would only find out one of the places that that protein could reside. So there are cell-wide methods that are based on protein correlation profiling, which will give you maps of the entire cell. And I'll go into more detail about how those work in a moment. But what you need to think about is that these methods have all got different strengths and weaknesses. Some methods will give you information on one protein. Some methods will give you information on the entire cell. There are various other mass spectrometry methods to do with using mass cytometry or imaging mass spectrometry, which will also give information at a different resolution. But what you need to think about for all of these is the resolution. And you're going to get the best resolution using microscopy, but also coverage. And you're going to get best coverage of the proteome and get more of a cell-wide map if you use protein correlation profiling methods. The bottom methods, because you're seeing the whole cell, is very sympathetic to proteins which are in multiple places, whereas it's not going to give you any information about cell-to-cell heterogeneity, where the live cell imaging methods, because you can look at cells individually, will give information about cell-to-cell heterogeneity. So I'll talk now about the protein correlation profiling methods, because this is what my lab's method is based on. So these methods are not new. They were first described by the Belgian cell biologist Christian de Duve many, many years ago. And what he postulated was that it's actually very difficult to purify any subcellular niche, so why bother? If instead you can somehow fractionate cells, and in those fractions you have your organelles spread across them based on some physical property, it doesn't really matter if each fraction is contaminated. What does matter is that each organelle has a specific pattern through these fractions. So this is the protein correlation profiling method. So let's imagine we fractionated our cells somehow, our organelles have stayed intact. The first fraction's got the lysosome in it and a bit of Golgi apparatus. Our second fraction has got more Golgi apparatus, less lysosome, and so on and so forth. If we then look at marker proteins and look at their abundance through these fractions, a lysosomal protein will peak in the first fraction, where a Golgi protein will peak in the second fraction, the ER, for example, in the third fraction, and so on and so forth. So as long as we've got markers that we trust that are present in those organelles, what we can now do is when we find a correlation profile of something that we don't know where it lives, we simply match it. So if it looked a bit like that, we would say, aha, we have discovered a novel ER protein. Maybe we have more of a composite pattern that looks like this. And here we might say, well, this protein is clearly in the endoplasmic reticulum, but it's also in the mitochondria. So in Christian de Duve's day, he had to measure all these correlation profiles using enzyme activity, which must have been extraordinarily tedious. But what we can do nowadays is to carry out the same sort of fractionation, but use quantitative proteomics methods to give us our correlation profiles. So the method that we developed many years ago now, we call LOPIT, or the localization of proteins using isotope tagging. We have 4 different versions of this now, soon to be 5. I'm only going to talk in the first instance at least about the 2 that are published, HyperLOPIT and LOPIT-DC. And the idea of both of them is that we subfractionate the cell in such a way that our organelles stay intact, and we can get these fractions which are then enriched in particular organelles. The HyperLOPIT method uses density centrifugation. So here our organelles are going to migrate through a density gradient, and we use a matrix called iodixonal, which is a self-generating gradient matrix, and our organelles will migrate within this matrix to a position which is consistent with their buoyant density. So the more dense organelles will end up at the bottom of the tube and the less dense ones at the top of the tube, and we can take fractions from these tubes which are going to be enriched but not pure for specific organelles, and we can use quantitative proteomics then to recapitulate the abundance of the organelles through these fractions. A simpler method is to use differential centrifugation. So here we're doing successive spins, taking the supernatant from one spin, spinning it again at a higher g-force until we end up with lots and lots of different fractions, with our last supernatant being enriched mostly for the cytosol. So the way that we crack open our cells is very important. If they are mammalian cells, we tend to use a ball-bearing homogenizer. And the importance here is to try and crack open the cells in such a way that your organelles stay intact. Because if they don't, then pretty much you've made a cell lysate and you're going to get no difference between the different organelle proteins. So either using density centrifugation or differential centrifugation, we get our fractions. We then use quantitative mass spectrometry. We can do this in multiple ways, either using tandem mass tags, which we tend to use, or using label-free methods. We get our different patterns for each protein. So each protein is going to give us a correlation profile, and then those correlation profiles, we use machine learning, and I'll explain a little bit about this in a moment, to work out patterns and which proteins are showing the same sorts of correlation profiles through our different samples, so through our different fractions. And what we end up with is something that looks a little bit like this. So this is Loppet data from a mouse embryonic stem cell. It was published a while ago. And what it is a principal component plot. So we have many dimensions because we'll be taking many different fractions in from our gradients or from our differential centrifugation. We get our quantitative data, and this is just reducing the complexity of our data into two principal components. But even from these two components, you can see that we've got clusters of proteins which seem to be doing the same thing. Now, the way to interpret these plots is a spot is a protein, and proteins that are coloured the same are either markers for a particular subcellular niche, or they've been classified to that subcellular niche using our machine learning tools. So the way we carried out this particular experiment, we have subnuclear resolution. In pink, we have proteins associated with chromatin, and in pale green, we have proteins that are associated with nucleoplasm. In blue down here, we have cytosolic proteins, and in this top left-hand corner, we have the secretory pathway from the endoplasmic reticulum and endosomes all the way to the plasma membrane. In red at the top, we have the mitochondria, and floating above it in the third dimension, you're going to have to believe me on this one, is the peroxisome. We can also get information on large protein complexes. In beige down here, we have components of the proteasome, and in the middle here, we have two clusters which correspond to the two ribosomal subunits. Now, half of the proteins here are in grey, and what they are proteins that don't classify to a particular location. They could be proteins in some cluster that we have no idea what it is because we don't have markers, so we can't give it a name. Or more likely, these proteins are proteins that are present in more than one place, and what we're seeing is the steady state location. So we're seeing the average. We're starting with millions of cells. And so if we have a protein down here somewhere, perhaps it's a protein that spends some of its time in the cytosol and some of its time in the nucleus. So we can also, from these data, have a look to see proteins perhaps in different isoforms, where those isoforms have got different subcellular locations. So the squares here are two different isoforms of leucine aminopeptidase. There's a long form with a mitochondrial import sequence, so that's mitochondrial, and a short form, which is in no man's land, so it's between the cytosol and the secretory pathway. If we look in the interactomics databases, what we find is that leucine aminopeptidase 3 has a variety of different interaction partners. But actually, when we look at the location of those interaction partners, we can see that the interactions of LAP3 are spatially restricted. So the long form interacts with these partners in the mitochondria, where the short form interacts with these partners in the cytosol and these partners in the secretory pathway. We can also map on large protein complexes, and it's interesting to see that most components of large protein complexes end up in the same part of the cell, but there are some exceptions to that rule. For example, this little red dot here, all its friends are over in the secretory pathway, where this looks to be potentially a protein which is involved in more than one complex because its steady state location is not consistent with just being part of a single complex over here. We, I don't know if this is going to play. We'll see. Oh, yes, it does. So we have developed a vast number of different tools, machine learning tools, and this has been the work in the past of Lorna and now Lisa Breckles, where we can take our data, we can visualize them in different ways, we can carry out different types of machine learning and clustering tools to work out what's there. And we try to make all our data publicly available. So Lisa's developed a variety of Shiny apps, which means that you can type in your favorite proteins and in our data set see where they are and what other proteins are within the same location on the plots. So we've had to develop various models and various systems and algorithms to be able to get as much out of our data as we possibly can. And what we're very concerned about is populating the literature and our databases with false discoveries. So we have carried out collaborations with mathematicians in Cambridge, particularly Paul Kirk. And Paul and I had a brilliant PhD student, Oliver Crook, who used Bayesian statistical methods to be able to determine the probability of proteins in different locations. So this protein, exportin 5, is a protein that shuttles between the nucleoplasm and the cytosol. And we need to know in our data what the proportions are of this protein in different places. So by using a TIG-augmented Gaussian mixture model, we're able to develop methodologies and get results which will give us some confidence and probabilities of proteins being in multiple places. So, having developed LOPIT, we can do two things with it. We can use it to get static maps of proteomes, or we can use it to get moving maps to see how proteins relocalize upon perturbation. So I'll give you an example of both of those. And the first was a collaboration, or still is a collaboration, with my colleague here in the biochemistry department, Ross Waller. And Ross is interested in Toxoplasma gondii, which is an obligate parasite. So it's a member of the Apicoplexa family, and it's pretty unpleasant. Its intermediate host is the cat, so cats carry this thing, but don't necessarily have any ill effects from it, where the definitive host is pretty much any warm-blooded animal, and quite a large proportion of the population are going to be infected with Toxoplasma, particularly those that are up and close and personal to cats, but it won't cause a problem unless you're immune suppressed. So anyway, very little was known about Toxoplasma in terms of its organelle components. It has a lot of organelles that you would expect a eukaryote cell to have, but some very specialized organelles whose job it is to enable the parasite to glide across the surface of cells and then to infect those cells, so to be taken into the cell in such a way that the immune system is evaded. So this is the work I'm showing here, which was published last year, of Konstantin Barilyuk. He was a very talented postdoc in Ross's lab. And what Konstantin did was to manage to crack open these cells in such a way that the organelles stayed intact, and then carried out the LOPIT protocol. And he was able to get exquisite resolution of all these very peculiar and unique organelles, which allowed Toxoplasma to do its job. So beautiful resolution of 26 different subcellular compartments. So now we know proteins that exist in these organelles that are very specialized, then these can be looked at in more detail to see if they would make reasonable therapeutic targets. And we can also look at the proteins in the different organelles and their evolutionary trajectories to see what proteins have evolved at which point in the apicomplexar family's transition from free living organisms to parasites. So that's an example of a nice static map, but ideally what we'd like to do also is to see how these maps change if we do something to the cell. So this we've been held up on for several years, mostly because of the informatics, not because of the way that we carry out the experiments, but what to do with the data afterwards. So we need to be able to see proteins that are moving from A to B. Maybe it's quite obvious that a protein starts off in the plasma membrane and ends up in the lysosome. Maybe proteins, when you perturb a system, don't move at all. But it also may be that a protein that was in multiple locations, when you perturb the system, is only in one, and vice versa. So we need to be able to map all those eventualities. And to be able to do this, we came up, or rather Oliver Crook came up with an algorithm, again, based on Bayesian statistics called Bandel. And the idea here is that not only are we coming up with a probability of a protein belonging to a particular subcellular niche, but also the probability of that protein moving upon perturbation. And what we end up with at the end of applying the Bandel algorithm is the rank order of proteins that are differentially localized between our experiments, so our different cellular conditions. So this has been applied in a fairly primitive form, it's much more advanced now, to a study that was published last month. So this study was trying to look at how proteins relocalize in response to initiation of the innate immune response. So we used a cell line, which is a monocytic cell line, and when you treat the cell line with lipopolysaccharide, then it starts to differentiate. It does this through activation of the toll-like receptor 4, and it differentiates to become an activated M1 macrophage. So we wanted to look at this process, and we were asking two questions. One was, can we capture the global changes in the spatial proteome? So we want to know what the cell-wide response of activation of this is pro-inflammatory phenotype. But we also wanted to ask a more fundamental question, which was, if you do something to cells, how much do they relocalize the proteome, or how much do they just use abundance changes? So if you can imagine, an awful lot of experiments have looked at how cells respond just by looking at abundance changes. So you do something to your cells, let's say you treat it with a drug, and you take control and you take treated, and you look to see how the proteins are changing in abundance. You might look how they change in post-translational modification. But very rarely do you look to see if the proteins are relocalized. So this is what we aimed to do. So this was the work of Claire Mulvey, a former postdoc in the lab, and she carried out both a spatial and temporal study. So she looked over a time course how proteins were changing in abundance. She chose the 12-hour time point to also look to see how proteins were moving around cells. The reason she did this is that at 12 hours, there were some major proteomics changes, but no particular phenotype changes. So she wanted to look at the early response. So having determined that 12 hours was a good time point, what she was able to do then is to collect her Lopit data, but she also had matching abundance data. And what she was able to show was that there was an awful lot of proteins that did move around, some from one place to another, some from multiple places to one place, from one place to multiple places, and so on and so forth. Interestingly, at the 12-hour time point, only 2 proteins changed in both abundance and location. So Lisa Breckles, who's an informatician in the lab, produced this wonderful alluvial plot or waterfall plot to show where proteins were moving to and from. And of the proteins that were moving around, 160 of them had not previously been implicated in this process. And if we were just looking at abundance changes, they still wouldn't be, because we wouldn't have picked them up. Many of these proteins are involved in nuclear cytoplasmic shuttling and reformatting of the plasma membrane and the cytoskeleton, so remodeling, which is the early signs of the phenotype of the cell changing. So just to reiterate this still further, I want to point out here in these two plots, so this is untreated and this is 12 hours after treatment, that there's a whole family of Rho GTPases that start off in the cytosol, move in a concerted fashion to the plasma membrane. But if we look at the abundance changes, and I'm just pulling out here CDC42, there are no abundance changes whatsoever during this process. But when we look at the Bandel plots, this protein is very much cytosolic in the unstimulated cells and very much plasma membrane in the stimulated cells. So this really is just to show, or to make the point, that if we're just looking at abundance changes, we don't know very much about what's actually happening in the cell and how the proteome is just being shunted around. Now, at 12 hours, we can't distinguish between the following. Is existing protein being moved from A to B? Or is protein at A being degraded and new protein being synthesized, which then traffics to B? So unless we do some sort of pulse SILAC and look at newly synthesized protein and where it ends up, these data don't distinguish between those two different eventualities. If we look at the proteins that are both changing in abundance and also in location, one protein, so CLEC11A, is a cytokine protein which ends up in the endoplasmic reticulum, and we think what this is, it's a protein which is secreted, so upon stimulation more of it's made, it's going through the secretory pathway, so that's what we're picking up. The second protein is part of the autophagy receptor, which makes some sort of sense in response to stimulation, more of it's made, and it is relocated to the plasma membrane. Okay, so that is the major point of the major part of my talk, which is to really talk about how we can chart how proteins are moving around the cell on a cell-wide scale, thousands of proteins per experiment in response to some perturbation. But we're also interested in the transcriptome and what role RNA plays in all these processes. So RNA is going to play two roles. It's going to be involved, of course, in the translation of the protein, but it's also going to be involved in binding to the protein. And it's known that RNA plays a big role in binding to the proteome in many ways, some of which, or many of which, are still to be determined. So what is the interplay of the transcriptome and the proteome? So the first thing we thought we'd have a look at is the RNA binding proteins. So RNA binding proteins are difficult to spot. Many of them, no, some of them, and you'll see the proportions of which in a moment, have got card-carrying RNA binding domains. But many others will bind RNA through short linear motifs, and lots of them, and they are difficult to predict. So we thought, to begin with, that we would use methodologies which exist to fish out the RNA binding proteome in our cells. And the way we thought that we would do this is to use the RNA interactome capture method, which has been pioneered by Matthias Henze and Marcus Lanthaler's labs. And the idea here is that you cross-link RNA to protein and then fish out what's been cross-linked using the poly-A tail polyadenylated messenger RNA. This is therefore very biased to proteins that bind to polyadenylated RNA. There are other methods that you can use, which involves synthesizing RNA with various non-standard bases that can be used in click chemistry. but it's very reliant on efficient metabolic labeling. So we thought that we would try and come up with a method that ultimately we could combine with our Lopit method to show where proteins are actually binding RNA in the cell. And we came up with a method that was much more sensitive and not biased to polyadenylated RNA. So rather than needing this many plates of cell culture to do an experiment, we need half of one of those. So this method we've called OOPS, or the orthogonal organic phase separation of RNA binding proteins. And it works in a really simple way. We published it in Nature Biotechnology a couple of years ago now, and there's a follow-on protocol. And what you do is to cross-link RNA and protein together using UV, and then you carry out the trisal extraction, which people have been using for years, with the idea that free RNA partitions to the aqueous phase and protein partitions to the organic phase. We argued that if RNA and protein are cross-linked to each other, there'll be one unholy battle between them, and they'll actually end up at the interface between the two phases. This is the very interface that the trisal protocol tells you to avoid. Well, we didn't avoid it. We took it. We carried out multiple different washes. And then we argued that what we'd end up with at this interface would be very, very enriched cross-linked protein and RNA. To work out the RNA component, we could then carry out protease treatment to digest the protein, redo the trisal extraction, and now our liberated RNA would go into the aqueous phase. Or we could do the reverse, treat the interfaces with RNAs, and now our liberated protein would go into the organic phase. And we could use mass spectrometry to work out what proteins were there in the organic phase, and next generation sequencing, RNA-seq, to work out what RNA had been liberated. So we could halve these interfaces and treat half with RNAs and half with proteinase K. And what we discovered was virtually all the transcriptome binds RNA. In fact, all the transcriptome binds protein. In fact, it will all bind protein. But in our method, very short transcripts we can't determine. Whereas a much smaller proportion of the proteome binds to RNA. I should also say that this method is not particularly efficient. So we're not going to pull out the entire proteome. But despite that, only a smaller proportion of the proteome binds RNA. So we discovered a whole lot of proteins using our method which are known to bind RNA, but a lot more that are RNA binding proteins that had not been classified as being RNA binding proteins. Because this is an easy method, we could try it out on lots of different cell lines to work out a sort of core RNA binding proteome. And we could have a look at the molecular functions of the proteins that we're now determining as novel RNA binding proteins. And interestingly, how many of them lack canonical RNA binding domains, which was quite a lot of them. We could also carry out judicious use of proteases such that we could end up isolating peptide with RNA bound by digesting the RNA down to a single base or so, and then use the methods that you use for purifying phosphorylated peptides to fish out our peptides with RNA bound, because peptides contain phosphate, and then sequence these and map them back to the structures. And what we found was that in many cases, the RNA binding sites on proteins overlap with features of interest. So in the case of PARP, so this is a nuclear protein involved in the DNA repair response. It's druggable. There are many drugs that have been designed to interfere with PARP's activity. But if we look where the RNA binding site is, it actually overlaps with the drug binding domain. And this is something that wasn't necessarily known, and it certainly casts in doubt how a protein in a cell will respond to a drug if its drug binding site is stuffed up with RNA. We also use this method dynamically by carrying out cell cycle arrest and release. And what we showed was that, again, in parallel to the dynamic Lopit story that I just told you about the LPS study, a similar sort of thing here, that our proteins were not changing at all in abundance during this process, but were changing in their RNA binding capacity. And this was particularly true for metabolic enzymes. So many metabolic enzymes we showed, and others have shown too, bind RNA. And when we had a look what happens during cell cycle arrest and release, these proteins don't change in abundance, but do change in their RNA binding capacity, which may be a moonlighting function, or maybe the RNA somehow is controlling their activity. And we were able to map, this is in the case of GAPDH, we were able to map that the RNA binding site in GAPDH is in the NAD binding domain, so in the Rossmann fold. All right, so we now have two methods. We've got OOPS, we've got LOPIT. The next thing to do is to combine them together to ask a different question now. So we know that we can work out what RNA binding proteins are present, but we want to know where those proteins are binding RNA in the cell and where they're not. So this is going back to potentially moonlighting functions, or at least putting some sort of functionality on subcellular location. So here we carry out, this is our density gradient. So we carry out LOPIT using density fractionation. We take out fractions, having cross-linked the proteins to the RNA before we start. And then the fractions we put through trisol separation. And the interface is going to give us the location of the proteins that are binding RNA. And the organic phase is the location of the proteins when they're not binding RNA. So therefore, we can get, for every condition, we can get two plots. So on the right-hand side, we've got the free protein, and on the left-hand side, we've got the RNA binding proteome, but the subcellular locations of these. So the example I'm showing here are tRNA ligases. So tRNA ligases are cytosolic when they're bound to RNA, and they're cytosolic when they don't bind RNA. So this isn't particularly unusual. This is what we would have expected. But if you look at these blue clusters here, this is what's in blue are proteins that are classified to the endoplasmic reticulum. And you can see that there are two subclusters in the RNA binding proteome, so what we call RBP map, so where the RNA binding proteins are in the cell. When we then have a look to see what proteins are in these subclusters, in the top one, these proteins bind RNA at the ER, and when they are not binding RNA, they're still at the ER. This bottom cluster are ER, so endoplasmic reticulum located proteins when they're binding RNA, but are largely cytosolic when they're not. And when we look to see what these proteins are, these are proteins that are involved in the cytoskeleton, are enriched in a particular domain of proteins that bind divalent cations. So this is certainly worth following up. But this, I'm not saying that these proteins necessarily moonlight, but it is showing that their RNA binding capacity is restricted spatially within the cell. So just finally, can we take our LOPIT method, where we on a cell-wide level can work out where all the proteins are in the cell, can we transfer this to the transcriptome? So can we also get a parallel map of where the transcripts are in the cell? So this is a method which we've called Lorna, the localization of RNA. And I'm not going to go into details, it's not published, but we've had to drastically change our subcellular fractionation methods. So we still use density gradients and we still use differential centrifugation, but we've had to tweak the method significantly, because if you don't do this, what happens is that your RNA, which are sort of the largest stringy molecules, tend to migrate to a position which is based on their size. And you don't want that. You want them to migrate to a position which is consistent with whichever organelle they're associated with. So now we take our gradients, we take our fractions, and we do trisol separation. And now the aqueous phase is going to give us a cell map of the transcriptome, where the organic phase is going to give us a cell map of the proteome. And so we should have two overlapping cell maps, and they come from the same fractions, so we can relate them to one another. So to show you that Lorna works, So this is a principal component plot of now the transcriptomes. This is RNA-seq data. There are fewer locations that RNA will be associated with, and we can work out pretty much what these are, and they make sense. We've got marker transcripts for the different organelles. We can use ternary plots to look at the data more straightforwardly. So here we've got cytosol, nucleus, and membrane, and you can see the distribution of the transcripts between these locations. Membrane here is compacting the ER and the mitochondria together. But it makes sense. We've got certainly transcripts that are shared between the membranes and the cytosol, and probably fewer transcripts that are shared between the cytosol and the nucleus. We can do a bit of sanity checking by looking at the location of transcripts that we expect to be in very much in one of these three places. And also then, because our methods are not biased to any particular type of RNA, we can have a look, for example, at the location of long non-coding RNA. And we were shocked to see that there is a larger proportion of long non-coding RNA in the cytosol. We've also, without going into any details, put spikes into all our fractions, so we can actually get hard numbers for the proportion of each transcript which is cytosolic and the proportion which is nuclear, for example. So we thought that we would use both Lorna and Loppet together to look at dynamic relocalization. So the way that we did this was to use thapsigargin, which is a protein, it's a drug which will elicit the unfolded protein response. And we showed that in the situation of the transcriptome, that what happens is that thapsigargin shuts down translation, and the transcripts that have been associated up here, this green cluster here is now endoplasmic reticulum, end up compacting back into something which is more cytosolic, but not every transcript does that. So I seem to be missing a slide, and I don't know why, but just to say that we end up with two sets of transcripts that were associated with the endoplasmic reticulum in untreated cells, but where we've driven the cells down into the unfolded protein response, there's a set of transcripts which are enriched in transcripts that go through a slightly different translation initiation mechanism, which are still ER associated or partially ER associated, and another set here which are enriched in transcripts which are thought to go into stress granules. So as we can see, transcripts, as I say, that are moving around the place. The vast majority of them don't. The nuclear ones are not affected, and a lot of the cytosolic ones are not affected as well. And as I said, one of the sets of transcripts that are still associated with the ER are enriched in transcripts whose initiation for translation go through a different mechanism involving EIF3D. If we look at the same data, but this time we're looking at the proteome, so this is the matching proteome data, we can now see sets of proteins that are moving around, and looking at the correlation profiles, we can see sets of proteins which are moving into the same fractions with the transcripts that are involved in stress granules. So the bottom line here is that we can identify new transcripts that go into these liquid-liquid phase separated stress granule droplets and the proteins that are going with them. So just to say that using this combination of methods, we can get matching maps of the transcriptome and how it's moving around in response to perturbation, and the proteome, and spatially how it is also moving around, and we can tie the two together. So in summary, I hope I've shown that mapping subcellular location and how it changes gives insight into function. I've shown that about half the proteome exists in more than one subcellular compartment. I've also shown a method which will give a very unbiased view of the RNA binding proteome and how we've revealed some unexpected RNA binding proteins that lack canonical RNA binding domains, that we can map the spatial transcriptome, and reveal the subcellular distribution of all RNA, including long non-coding RNA. And then in a single experiment, we can capture both a map of the proteome and a map of the transcriptome and how this changes on perturbation. And I hope that these methods will be very useful to the community and will give essential insight into mislocalization and aberrant trafficking. So finally, I need to thank all my collaborators, particularly Anne Willis's group, who we've worked tirelessly with to come up with the OOPS protocol and also the Lorna protocol, and of course our funders. And here is in the UK, I'm sure it's the same in Brazil, we now have to have hybrid lab meetings where we all wear masks, and right at the back are our lab members who are zooming in. So thank you very much for your attention, and I look forward to any questions which are going to be filtered through Alexandra.
Well, thank you, Kathryn, for your great talk. Very interesting results. And so let me see in the chat. Well, just a minute. Pessoal do suporte, eu consigo ver as perguntas aqui?
Alexandre, sim, se você clicar em perguntas, vai estar lá as perguntas. Olha lá, apareceu uma.
Okay, we got. Okay, well, the first question is from Giuseppe Palmisano. Thanks for coming. Dear Kathryn, thanks for your wonderful presentation. I would like to ask, what is the state of the art on large-scale methods to map 3D structural changes associated to subcellular localization?
That's an interesting question. I'm not sure what state of the art is. I mean, if you can purify a protein from a particular subcellular niche, then cryo-EM, you know, the structural methods for working out on a more, I suppose, in vivo scale. Nobody's really gone to town on this yet, but you might imagine that you could use perhaps some of these limited proteolysis methods, so that you would use proteases on proteins that have not been unfolded or reduced, and you're only going to be able to make proteolytic digestions in peptide bonds that are available. So this is Paolo Picotti's work in Zurich, and if you started to look at different subcellular locations, maybe that you would have different peptide bonds available for proteolysis. And if you map that back to structures, it may tell you perhaps what's exposed in one location compared with another.
Okay. Well, many people saying thanks for the presentation, for the very nice presentation. Sergio Schenkman asks, very nice presentation. I would like to ask how you consider the intracellular phase separation after cell disruption. Is it applied to your technique?
Well, we were surprised, very surprised, and this is only the work that has come to light in the last few months. In our Thapsigargin treated samples, that we were maintaining stress granules within our gradients. So what we did was to use one of the sort of the markers for stress granules, G3PB1, and it was tagged. We tagged it with GFP. And so we could then look using microscopy at each fraction to see if we could see these granules, and they were there. So the granules bizarrely survived the ball bearing homogenization. So they didn't coalesce or they didn't disappear. So I think our methods, I don't we haven't tried this with the differential centrifugation method, but certainly with the density centrifugation method, the granules survive. So we do see them. And this is why we've got very unique correlation profiles for those granules for both the transcriptome and also the proteome.
Okay. Fabio Nogueira asking, thanks, Kathryn, amazing work. Is it possible to associate the subcellular localization with PTMs, for example, phosphorylation using LOPIT?
Yes, this is work that I didn't have time to present, and it's quite preliminary for us, but you can take the fractions and then digest the peptides or whatever you want to do, and use titanium dioxide. In our case, this is what we did. We used titanium dioxide to fish out the phosphopeptides, and then you've got two overlapping maps again. You've got as long as you do it after fractionation, then and actually you can do it after TMT tagging, but as long as you've done it after fractionation, you've got two maps. You've got the maps of the proteome and you've got maps of phosphopeptides, and so you map those phosphopeptides back to where the protein is. We went one step further and we tried to look at the location of within the correlation profiles of the unphosphorylated version of a peptide and the phosphorylated version. And what we found was in a lot of cases, it makes no difference whether the protein's phosphorylated or not, it's in the same place. But in some cases, we could see differences for particular phosphorylation events where we saw very different locations for the phosphorylated peptides. We also, because we had an interest in metabolism for a completely different project, used a method to pull out proteins that was peptides that were succinylated. And there we were able to show if a protein succinylated, which can happen non-enzymatically because of high levels of succinyl-CoA, we could see the location of the succinylated peptide. And we imagined that this would, because we were very naive when we started out, we imagined that that a succinylated protein would be largely mitochondrial because that's where succinyl-CoA is made. But what we showed was that actually succinylated peptides are in a lot of parts of the cell, not everywhere, but not as much in the mitochondria, because in the mitochondria there's a desuccinylase, SIRT5, that comes along and takes the succinylated groups back off again. So that was a long answer, and the bottom line is yes.
Okay. Maria Juliano said, Kathryn, thank you so much for the beautiful presentation. Adam Minas asked, how this can be applied in tissues?
LOPIT we've done in tissues before. We've even done it on Drosophila embryos. So for tissue, it's fine, and therefore I imagine also that, although we haven't tried it, that Lorna would be as well. The problem with the RNA binding proteome and tissues is that we need to UV crosslink, and that's fine for cells in culture. It's even fine for anything that we can sort of spin down and spread out. But if you had a large chunk of tissue, the UV wouldn't penetrate evenly. So I think there is a limit for looking at the RNA binding proteome in thicker sections of tissue. If it's a very thin section of tissue, then it may be okay, but we haven't tried it.
Okay, Dulce Casarini said, thank you for the fantastic presentation. Studies presented here with different proteomics techniques, including real-time localization of protein movement in a cell, must have a very high cost per analysis. What is the laboratory access to the technology presented here and the approximate cost per analysis?
For the analysis, by analysis, if you mean mass spectrometric analysis, then that depends on whatever a facility charges. For LOPIT, we would end up with, say, 12 fractions. Plus, if we use a TMT workflow, in fact, if we don't, it's still about the same. So that's whatever would be the cost of 12 runs on a mass spectrometer. For RNA-seq, for the Lorna method, we spent £20,000 on one experiment. But it was the first experiment we did, and we sequenced pretty deeply. So my understanding of RNA-seq, which I have to confess is a fairly new field for me. I mean, we've not ever used it much in my lab before the last couple of years. I think you get what you pay for, and we've only ever used Illumina sequencing. I know some other sequencing platforms may be cheaper, but it depends on the depth of coverage that you require. In terms of analysis, as in data, the cost there, well, I suppose the cost is time. They're not massive data sets to store, but it can take a while to analyze, and you do need to have good sets of markers for each organelle, for both the protein and also for the transcripts. So I suppose that can be costly in terms of time. I'm not sure if that answers the question, but yes, I mean, these are not necessarily cheap experiments to do, but the data you get at the end is so rich that it's impossible for one group, I think, once you have these data, to really mine them to their full, which is why it's important, I think, to make good open source tools for other people to go in and be able to interrogate the data.
Okay. Professor Luis Juliano asked, do you consider to explore mapping protein-protein interactions?
No, we could do. I mean, we could try and pull complexes out of different locations and see if the components change. It can be done, but we haven't really explored that very far. We have other methods in the lab that I didn't talk about to pull out complexes from membranes that we would then make a very enriched membrane prep and go in looking for particular complexes. But we haven't done this on sort of lots of different Lopit fractions.
Okay, another question from Joseph Palmisano regarding the T-Gond work. Do you think that the subcellular localization of unannotated proteins could give some clues on these proteins?
We hope so. Yes. For the ones that are completely unannotated, if we know where they are, then that would give some idea of the processes that they're involved with. And it may be that you then go in and target them in a different way, either pulling them out to see what other proteins they are associated with. For proteins that are in more than one location, where we think we know what the protein does, then I think these are really fascinating, because why is the protein, if we think that this protein is a mitochondrial protein and this is where it does its job, if we also find it somewhere else, then what's it doing there? I mean, all these processes that I've described, these cellular processes, are expensive for the cell. So I don't think trafficking proteins to multiple places is done by accident. So there must be another reason why a protein ends up in a different part of the cell. So this is why I think looking as much as you can at trying to combine these data with functionality, like we did with RNA binding functions of proteins, and see if that's spatially restricted, might give insights into the fact that some proteins are doing different jobs in different parts of the cell.
Okay, Luis Juliano asked it again. He says, he means protein-protein interactions in side cells, I think single cells maybe.
In single cells, our methods are many things, but they're not single cell. So we can't, we start with millions of cells. So how people would, protein-protein interactions using microscopy methods, I suppose, is something which there's lots of groups that have looked at that. In fact, there's a very nice paper in Nature from Emma Lundberg and Trey Eidecker, I might have pronounced that wrong, looking at comparing or combining IP data, so pulling down protein complexes with microscopy data and using machine learning tools to really delve into that more heavily. But in terms of the methods that we've developed in my lab, they're not single cell methods.
Okay, from Adriana. Thanks, Kathryn, for the nice presentation. Do you think it's feasible to use spatial proteomics combined with native co-fractionation plus MS to evaluate protein-protein interaction?
I would imagine, I'm trying to get my head around these two different methods. So groups like Ed Marcotte, Andrew Emily, and Leonard Foster have used co-fractionation a lot on complexes. And what you could do is to fractionate, and then you'd sort of then have a three-dimensional matrix, because if you fractionated and then for each fraction did more co-fractionation, then that's something that could be done. The issue would be the sensitivity of the system and the losses of material. So by the time you've done this, I would think in the case of a lot of fractions, there's not enough proteins within those fractions to then do yet more complex fractionation. So I think for the more abundant complexes, this is probably the case. But the data analysis at the back end of it is going to be fiendish, because you have got this three-dimensional matrix now. So if you fractionate, you're doing co-fractionation of a particular complex in the nucleus that you think is that complex in the nucleus, and for example, the same complex in the cytosol, you are going to have contamination of each. So you'll have some cytosolic complex in the nuclear fraction and some nuclear complex in the cytosolic fraction of the same complex that's in these two different places. So you've got to work out the correlation of the proteins within the fractions, and then the correlation of the complexes when you try and fractionate them away from other complexes in the same fraction. So I'm sure that it is achievable, but I think it's tricky. It will be a very tricky experiment, and informatically, it's going to be a tough ask. Not impossible.
Okay, I think we have no more questions here in the chat. I have one question, Kathryn. For example, now that we have more options of TMT tags like 16 or maybe 20 tags, would it be compatible with the number of fractions that you can achieve with the centrifugation, or would we be able to fractionate more organelles?
We've done quite a lot of work on this and spent vast amounts of money on these tags. So the more fractions you have, the better the resolution, up until a point. But actually, I think the best use of these tags, where you've got lots of them, is to combine two different cell states in the same labeling. So if you were, for example, to use the 18-plex TMT, you could have 9 fractions from untreated cells and 9 fractions from treated cells. And then the good thing about doing this is you're cutting down on mass spectrometry time, of course, but you're also removing a lot of technical variability from downstream the experiment. So we have done this where we've got, and we've shown it, although we've not published it, where you've actually got much tighter movement data if you're carrying out 2 experiments in the same plexing. So 9 plus 9 if it was I think we did 8 plus 8 when it was 16 plex.
Okay, nice, Kathryn. Well, let me see if we have more questions. I think no more questions. Well, Kathryn, thank you so much for your great talk. It was so many interesting results and so many things to think about of quantitation versus subcellular localization. And well, I hope we can meet next time here in person at the Federal University of São Paulo. And again, thank you so much for your talk.
You're welcome. I believe I've stopped sharing. Can you still hear me?
Yes.
Yeah, I think I've stopped sharing. You're very welcome.
Okay, so thank you again. With this, we finish this webinar, and thank you for the public for coming too, and thank you for the support team. Okay, thank you again, Kathryn.
Okay.
Okay, bye.
Bye.